We are standing on the edge of a monumental shift in human history, the moment we gain the ability to permanently alter our own genetic code. And the year 2026? Well, that's shaping up to be a critical flashpoint where this whole debate stops being theoretical and gets very, very real. We're not just talking about curing diseases anymore. We are on the verge of being able to design our descendants. So let's unpack what that really means.
So here's the core issue, right? There's this incredible tension. On one hand, pretty much everyone agrees the technology is just not safe enough for the clinic yet. But on the other, there's this really strong push from inside the scientific community for what they call a translational pathway. And all that really means is a roadmap, a way to get this technology from the research bench to actual human use. This isn't science fiction. This is happening now in labs and policy meetings. And that's what makes this so urgent. The question has totally shifted from if we'll do this to when and how. You have private companies like Manhattan Genomics already announcing plans to correct genetic mutations in embryos. That means the ethical line in the sand we've all been talking about is being approached right now. Our reality check for 2026 has never been more important. Okay, so to really get your head around what's at stake here, we have to talk about the two different paths for gene editing. It's somatic versus germline. And trust me, this one distinction is the absolute key to understanding the entire debate. Think of it like this. Somatic therapy is like finding a typo in a single printed copy of a book. You take out a pen, you fix it in that one book, and that's great for that one reader. But every other copy is unchanged. Now, germline therapy, that's like going back to the original manuscript, to the master file, and changing it there. Every single copy of that book, printed from that point on, forever, will have that change. You're not just editing one person, you are editing the future of the human story. And the revolutionary tool making all this possible is CRISPR-Cas9. You've probably heard it called molecular scissors, and that's a perfect analogy. You have a guide molecule that's like a GPS, taking the scissors to the exact spot in our DNA, and then an enzyme makes the cut. What's so wild is how precise and relatively easy it is to use. It has completely supercharged genetic research and dragged these massive ethical questions out of the lab and right onto our doorstep.
So you're probably wondering, is this stuff even legal? Well, the short answer is, it's a mess. There's no single unified rulebook. What we have is a world of contradictions. Fragmented is the word experts keep using, and it's spot on. It's a patchwork of laws that are all over the place. What's completely banned in one country might be legal for research just across the border. It's confusing, it's inconsistent, and it makes having a global conversation incredibly difficult. So on one end of the spectrum, you get really clear lines drawn. The Oviedo Convention, for example, is about as direct as it gets. It basically says you cannot make any change to the human genome that can be inherited by future generations. Full stop. No exceptions. For them, the human germline is sacred and totally off limits. And this table just illustrates that fragmentation perfectly. Look at the European Union. As a whole, it bans clinical trials that would edit the germline. But then you look at France, a member of the EU, and they now specifically allow research on embryos, as long as it's under really strict conditions. Meanwhile, in the US, there's a federal ban on making a baby this way, but the whole regulatory system is super complex, split between different agencies. The big takeaway here, there is no global consensus. Not even close. Now, all this caution isn't just about abstract philosophical debates. It is deeply rooted in some very real and very serious biological risks. Let's look at the scientific challenges, the big unknowns, that give even the most enthusiastic supporters of this technology a reason to pause. The first huge risk is something called off-target effects. So remember those molecular scissors? They're incredibly precise, but they're not perfect. Every once in a while, they might cut a piece of DNA that looks a lot like the target, but isn't. And that creates an unintended random mutation. Now, in regular gene therapy, that's a problem for one person. But in germline editing, that one little mistake becomes a permanent, heritable genetic error that gets passed down to all future generations. And the second major hurdle is something called genetic mosaicism. This happens when the gene edit doesn't take effect in every single cell of that tiny early embryo. What you end up with is a mosaic, a person who is a mix of both edited and unedited cells. This makes it basically impossible to know if the therapy even worked, and it could mean the original genetic disease is still lurking in some cells, leading to totally unpredictable outcomes down the line. And look, these aren't just theoretical what-ifs. The scientific literature is absolutely filled with studies that detail these exact risks.
Off-target cuts and mosaicism are the two biggest reasons why the global scientific community is in agreement that we are just not ready to try and make a baby with an edited embryo. The risks are just too high and the consequences are way too permanent.
So let's just imagine for a second that we could solve all of these biological problems. Let's say the tech becomes 100% safe and effective. Well, that's when we arrive at the ultimate choice, the real beating heart of this entire ethical debate, where we draw the line between therapy and enhancement.
This is it. This is the big one. Are we using this unbelievable power to heal heartbreaking diseases, or are we cracking open the door to designing our own children? Because that line between treating a disease and enhancing a person is razor thin and incredibly blurry.
So what do we even mean when we say enhancement? The key difference is the goal. Therapy is about restoring a person to a normal, healthy baseline. It's about fixing something that's broken. Enhancement, on the other hand, is about trying to improve a person's abilities beyond that normal baseline. It's not about fixing a faulty gene, it's about swapping a perfectly good gene for one that's considered better.
And the ethical objections to this are just profound. First, you're making choices for a child who can't consent, potentially limiting their future. You risk changing the parent-child relationship from one of unconditional love to one of, well, production. And you can just imagine a world where this creates a new kind of social inequality, a genetic divide between the enhanced and the unenhanced. Finally, it just speaks to the strive to master and control every aspect of human nature, leaving no room for chance or natural gifts.
And this brings us right to the famous slippery slope argument, which the Council of Europe sums up perfectly here. The big concern is that even if we start with the purest intentions, only using this tech to cure horrible diseases, we will inevitably open the door to enhancement. Once the technology is accepted for therapy, the social pressure to use it for improvement might just become impossible to resist.
So in the end, as we stand here looking toward 2026, it all comes down to this. The human gene pool isn't just my DNA or your DNA. It's our shared inheritance, the product of millions of years of evolution that belongs to all of us. And as this incredible, powerful technology comes into focus, the biggest question we have to ask ourselves is a pretty simple one. Who gets to be the editor?